This is a talk I gave at the BioInfect conference on 26 November 2013 in Alderley Edge, UK. I will describe two examples of antibiotic resistance in bacteria isolated from animals. The first is fluoroquinolone resistance in Campylobacter, and the second is extended spectrum beta-lactam resistance in Enterobacteriaceae. Before I do, it's important to remember how antibiotics are used in animals reared for food production, particularly those where animals are reared intensively, as shown in these photographs. In these circumstances, where one or more animals becomes ill, antibiotics are administered to the entire flock or herd to treat the infection and to prevent the infection spreading and having a large economic impact. So how do bacteria become resistant? Well, the first example I will give of fluoroquinolone resistance is a very good example of Darwinian evolution, where a resistant mutant is selected up for an environment. So for example here in the left-hand circle there are blue and red dots. The blue indicates the antibiotic susceptible bacteria and the red the resistant. If this is a petri dish or a human body or an animal, then in the presence of an antibiotic, all the antibiotic susceptible bacteria are killed, leaving only the antibiotic resistant bacteria able to grow. Because bacteria grow so quickly, overnight this single antibiotic resistant bacterium multiplies greatly to become a very large number, which can then proliferate and spread. So the example of chromosomal mediated resistance is that of ciprofloxacin resistant Campylobacter. This is some old data from the UK, but it shows that soon after the introduction of fluoroquinolones into veterinary use, such as enrofloxacin and sarofloxacin, fluoroquinolone resistant Campylobacter were isolated from people, and that over a few years the numbers of these increased rapidly. Data as recently as 2011 shows that there are a large number of bacteriologically confirmed Campylobacter infections in people, but these are predicted to only be about 10% of the actual number, because most people with gastroenteritis are not so ill that they visit a doctor and give a sample for bacterial analysis. As of 2011, approximately 15% of Campylobacter isolated from people were resistant to fluoroquinolones. And if one multiplies up the anticipated number, this gives about 97,000 resistant infections in the UK alone. It is well recognised that Campylobacter is a zoonosis and enters the food chain via a number of sources, but in particular poultry. Nonetheless, data such as this in the 1990s was taken only to be an indication of what may be happening, and there was no clear evidence that the resistant bacteria had arisen in animals and transmitted through the food chain to people. Therefore, in a DEFRA-funded study, we worked closely with food producers and veterinary surgeons to identify commercial poultry flocks that would be receiving an antibiotic, and that we could go in and sample feces from the poultry houses and examine them for the presence of bacteria and resistance. As you can see on this chart here, the clear white bars show feces in which there were no resistant Campylobacter, and the grey bars show those in which there were resistant strains. It is very clear to see that resistant strains were very rare before treatment, but during treatment they dominated all samples, and indeed after treatment, when there was no longer any antibiotic present, and up to 4 weeks until the birds were slaughtered and entered the food chain, these bacteria persisted in most samples. This is clear, unequivocal evidence that fluoroquinolone resistant bacteria were selected during exposure of birds to fluoroquinolones, and these indeed did enter the food chain. So what are the consequences of fluoroquinolone resistance? Well, the bacterium changes itself in exactly the same way, whether the resistance has come through the food chain or in people, so it's the same mutation. The same types of bacteria were found in animals as in humans. And data from a variety of sources has shown that fluoroquinolone-resistant Campylobacter are much more difficult to eradicate from poultry, but also increase the duration of illness in people. So there are other consequences of the antibiotic resistance. And of course, treatment of people, should it be required, will take much more time because the original drugs will not work and a second choice must be sought. Of course, the same is not true for all antibiotics, and we did another study on several different agents, and I've just shown here tetracycline, and shown that prior to treatment with chlortetracycline, again, occasionally antibiotic-resistant bacteria were found, but that these did not proliferate during treatment and did not persist post-treatment. So what was true for fluoroquinolones was not true for chlortetracyclines, so one should be careful about drawing broad conclusions for one antibiotic to others. The second example is of transmissible resistance, of where bacteria can share their genes and transmit them from one to the other, as shown on the left-hand side of this slide, showing a tube going from one bacterial cell to another, and through this tube, via a process called conjugation, antibiotic resistance genes are transferred. The example I will describe is that of the extended spectrum beta-lactamase CTXN14, and it's been known for a number of years that this enzyme, shown here in pale green, is disseminated throughout the world and has been detected in numerous different samples from people. In 2006 was the first description of this enzyme in a bacterium isolated from an animal in the UK. It was found in calf samples and a variety of other samples on this farm, and the enzyme encoding CTXN14 was found on a common plasmid which we call PCT. And this plasmid was found in many different strains of E. coli, and laboratory experiments showed that it could transfer very efficiently to new bacterial hosts. We sequenced the genome of this transmissible element and identified 6 regions to make a new test to look for PCT-like elements in samples. We received samples from all over the world, and we were able to show that there were PCT-like elements in Australia, endemic in Spain, in hospitals, farm animals and the environment. But more importantly, the first ever description of CTXN14 was encoded on a PCT-like element. We went on and used this test both at the VLA and HPA and showed that PCT-like plasmids could commonly be isolated from E. coli in people or animals. So what made this plasmid so successful and transfer around the world? Well, the first thing we thought of was that it must be widespread use of the antibiotic. And we used molecular tools to inactivate genes on that plasmid, and we showed that the absence of antibiotic pressure and the absence of the CTXN14 beta-lactamase had no effect on its persistence or biology. Therefore, there's something else on this plasmid that's allowed it to become so widespread throughout the world. And the presence of the antibiotic has then allowed this to cause a problem. So in conclusion, controlling antibiotic-resistant organisms depends upon the mechanism, whether it's transferable to bacteria or only to daughter cells, whether there's several genes on one element or is mediated by a single genetic event, and the antibiotic, the resistance mechanism and the species all play a role in controlling antibiotic resistance. But it's important to note that until proven otherwise, the persistence of antibiotic-resistant bacteria or these transmissible elements should be expected even when there are no antibiotics, and that this will not be resolved by good antibiotic stewardship in veterinary or human medicine. Therefore, we need to increase our understanding of the biology of resistance, and until we understand this properly, find other means to reduce resistant bacteria. Thank you.